welcome back, and a quick recap of what we have been talking about. We started talking about the patterning of embryo or embryo patterning. Embryo patterning is when the cells which are involved in the formation of embryo, they acquire a specific structure and function, and this is what we call as embryo patterning. One division each in the apical and a basal cell of the two-celled pro-embryo which you see here result in tetrad stage of pro-embryo. You can see that there are 4 cells which are formed by one division each in apical and basal cell, and this 4-celled structure which is formed undergoes yet another division to form an octant configuration. The basal cell, it divides asymmetrically and transversely, forming the suspensor. So, you have to remember that basal cell is primarily only forming the suspensor. In rare cases, it may give rise to root or root-like structures, but otherwise it will be forming the suspensor only. The apical cell can undergo transverse or longitudinal divisions. In figure A, you see that the apical cell undergoes a transverse division, and we can see a linear row of 4 cells. Okay. So, if the apical cell, instead of undergoing a transverse division, it undergoes a longitudinal division. So, you see in the lower two figures, the apical cell undergoes a longitudinal division. So, in subsequent 4-celled pro-embryo, 2 configurations are possible which you see in A, that is a linear configuration, and a T-shaped configuration in the rest 2, that is in B and C diagrams. In the linear tetrad, 2 daughter cells L and L dash of the apical cell undergo 2 vertical divisions at right angles to each other, and they form an octant. Octant is comprising of 8-celled. This 8-celled stage has 2 tiers of 4 cells each, which you can see here. You can see that 2 tiers have been put separately on the right hand side to give you an idea that there are 2 layers basically of 4 cells each. In T-shaped pro-embryo, T-shaped is formed when the apical cell undergoes a longitudinal division instead of a transverse division. Here again there is an octant with 2 tiers of 4 cells each which is formed, and this is formed after 2 rounds, and after 2 rounds of division, that is 1 longitudinal and 1 transverse, again there is an octant which is formed. So, there are 2 tiers of 4 cells each. Third type of pattern is where all the 8 cells are in one tier, okay, and they are surrounded by 4 peripheral cells. Both the types of octants occur in monocots as well as dicots. Thus, at the 8-cell stage, the pro-embryo consists of an upper and a lower tier of 4 cells each and a suspensor. These 3 domains, now there are 3 domains which are formed, the upper tier, the lower tier and the suspensor. They remain fixed and they go on to form the organogenetic parts of a mature embryo. Now, you would recollect in the last lecture we discussed that first division in the zygote is transverse and asymmetrical. So, it results in the formation of a larger basal cell and a smaller apical cell. Apical cell divides longitudinally and each of the 2 cells divides again transversely, resulting in a quadrant. Another division occurs in each of the cells and it results in an octant, and this octant has 2 tiers of 4 cells each. The upper tier, the apical tier, is the apical embryo domain, and the lower tier, the basal tier, is the central embryo domain. The cells of the octant proembryo, they divide periclinally. You know periclinally is when it is parallel to the surface. So, they form a 16 cell globular embryo resulting in the establishment of a radial axis. This globular 16 celled proembryo now comprises of an outer layer. Outer layer has been formed by the periclinal divisions, and of course there is an inner layer of 8 cells each. Radial patterning leads to the formation of now 3 tissue systems of the plant. The outermost is the dermal, which forms the protoderm, which is a precursor of the epidermis. And then we have the ground and the vascular tissues which are formed. Longitudinal divisions in the inner core, they produce a central mass of proembryo. Upper tier cells, they form the shoot meristem and the cotyledons, whereas the lower tier cells form the vascular tissue, ground tissue of stem and root. That is how the embryo patterning happens. And now you can understand that there are different cells, there are different tiers of cells which are assigned to perform different functions. You can see in this particular one, this is a picture taken from a paper published in Nature. This is embryo patterning or organogenesis which happens in the embryo. So, there is of course a zygote which divides into 2 cells, the basal and the apical cell. The basal cell always undergoes transverse divisions, or it may decide not to undergo any of the divisions, and this will eventually form a suspensor. The apical cell, by undergoing either transverse or longitudinal divisions, it increases the number of cells which are formed, moving from 2 celled to 4 celled to 8 celled octant and then 16 celled proembryo, wherein 3 different layers are formed, the outermost or the dermatogen giving rise to the epidermis, whereas the other 2 which are in the central portion of this proembryo, they give rise to the shoot apical structure and of course the root structures, and you can see that there is a differentiation which is happening in the cells of the embryo proper here, and this is what is embryo patterning. And of course, this embryo, as the number of cells increase, it moves from a 16 celled to maybe 32 celled structure, and from a globular to a heart shaped or a torpedo staged embryo. Embryogenesis in monocots is slightly different from what we observe in dicots. Naias lacerata is a monocot. Here the first division in the zygote forms 2 daughter cells, a smaller apical cell and a larger basal cell. You can see here that basal cell is not undergoing any further divisions, and it matures into a single celled suspensor haustorium. Apical cell again divides transversely, building a filamentous row of 4 cells, and all these cells contribute to the formation of embryo. Periclinal divisions are parallel to the surface, in contrast to anticlinal divisions which we sometimes refer to where the division is perpendicular. Okay. So, periclinal divisions add layers, whereas anticlinal divisions, they add the girth. Periclinal divisions in 4 celled tier result in an octant tier. 4 axial cells are surrounded by 4 peripheral cells. These peripheral cells, they form the protoderm. Now the embryo is slightly globular in shape. You can see the first diagram over here. 3 of the 4 axial cells, they show pronounced divisions, and the 4th remains mostly undivided. So, there are unequal divisions which are occurring among the axial cells. This disrupts the symmetry and it results in an embryo being humped or notched, which you can see in the two drawings lower down on the right hand side. The notched part of the embryo is the cotyledon. So, there is only one cotyledon formed in monocots. Now, if we have to differentiate between dicot embryogeny and embryo and monocot embryogeny and embryo, in dicots, the two opposite cells of the terminal tier of octant proembryo contribute to the formation of cotyledons. In monocots, there are a variable number of cells of the octant proembryo which give rise to just one cotyledon. In dicots, development can be divided into globular, heart, torpedo or walking stick stages, and in monocots there is only the globular stage which is seen. In dicots, during transition from globular to heart shape, bilateral symmetry appears due to equal growth of cotyledons. Whereas in monocots, there is a rapid growth of only one cotyledon. In dicots, the epicotyle and hypocotyle both are terminal structures. Okay. In monocots, the epicotyle of course is a terminal structure, but hypocotyle is a lateral structure. Now, as discussed earlier, in dicots, mature embryo has two cotyledons, whereas a mature embryo in monocots has only a single cotyledon. Now, let me tell you that suspensor is a group of cells which pushes the embryo proper into the endosperm cavity and it connects to the surrounding maternal tissues. This suspensor can vary in shape, size, number and ploidy of cells. It may be highly reduced in size as seen in bryonia, or it may even be absent in case of viola. Suspensor can be single celled, and if it is single celled, it can be sac like or it can be tube like, or it can be filamentous in a single tier, or it can be even multicellular or clustered. The number of cells can be as high as 200 as in Phaseolus, or they can be only up to 20 in Brassicaceae, but you know the extreme is when it is only single celled. A well-defined suspensor is seen in all those families where there is no endosperm to nourish the embryo. And if you will recall, Orchidaceae, Trapezae, Podostimaceae are the families where there is no endosperm which is present. So, how does the embryo derive its nourishment? So, one is that of course there is a well defined suspensor which exists in Orchidaceae and Podostimaceae. Here you can see on the left hand side, that is the A picture, this is an example of orchids. Suspensor can be single celled or it can even be a multicellular structure. So, the four drawings on the left hand are of the orchids. In Podostimaceae, that is the B diagram, the suspensor is well developed and this basal cell, it grows hostorial branches also which penetrate the different parts of the ovules and they try to take up the nutrition from there and pass it on to the developing embryo. The C1 on the lower left side is an example of family Rubiaceae. Initially the suspensor is filamentous, but later it shows some lateral protrusions. The D or the fourth one is Crassulaceae, where the suspensor is not formed by the basal cell, but it is formed by the apical cell. In Crassulaceae, the apical cell forms a four-celled suspensor. Suspensor generally develops from a basal cell. Rarely it develops from the apical cell. Like the apical cell divides and the daughter cells, they contribute to suspensor, like we saw here in this particular diagram, that is in Crassulaceae. Suspensor is a fast growing structure, but this structure is very short lived. Mostly it exists when the embryo is transitioning from the globular to the heart shaped stage of pro-embryo. Probably it is the time when it requires the most nutrition. Suspensor has an absorptive behavior and it plays a role in nutrition of the embryo. Besides providing nourishment to the embryo, it also regulates the development of the embryo. Now, what is the ultrastructure of a suspensor? Ultrastructural specialization is seen in either heart or the globular stage of the pro-embryo, and the first step towards the specialization is the initiation of wall invaginations or wall infolds, which can appear either in outer or inner walls of the suspensor cells. Now you know that wall ingrowths, if there is wall ingrowth, it leads to an increase in the absorptive surface. Okay. Now along with the wall ingrowths, there is also an increase in number of ribosomes, plastids, mitochondria and other organelles which are present in the cells of suspensor. And this increase is followed by their arrangement in rafts parallel to the wall invaginations, supporting the transverse cell like activity of hostorial cells. Here you see that this is an ultrastructure of suspensor. Diagrammatic representation of ultrastructure of the hostorial cell of suspensor. You can see that there are wall ingrowths. Now, if you look at the diagram carefully, you can see that towards the lower portion of this cell there is some dark black type of coloration, and these are what are representing the wall ingrowths. Okay. Most of the cell organelles are also concentrated at the micropylar region of the cell. The cells of the suspensor along with hostoria, they remain connected with numerous plasmodesmata. You have to remember here that suspensor is a cell which is involved in uptaking nutrition from the adjacent or the adjoining cells, but once it has taken up the nourishment or the nutrition, it has to be passed on to the developing or the growing embryo. And how does it do that? Of course, it is by numerous plasmodesmata which are present between the suspensor and the other hostorial cells which are there. Role of suspensor, of course you would have understood by now that it serves as a conduit for nutrients and growth regulators which are required for embryo development. So, the primary function of the suspensor is to take up nutrients or growth regulators from the cells which are present around the embryo and then pass them on to the embryo for its growth and development. The position and structure of the suspensor also indicates that it has a possible role during embryogenesis, probably because it pushes the embryo proper into the endosperm cavity and connects it to the surrounding maternal and endosperm tissues. The suspensor has the ability to transfer molecules due to presence of cellular structures. You know, these cellular structures are like the wall ingrowths, the hostorial outgrowths, and of course numerous plasmodesmata which are present. Suspensor can serve as a channel for exchange of nutrients, growth regulators between the mother plant and the embryo. Additionally, the suspensor facilitates direct communication with adjacent seed tissues. You know, seed tissues can be a seed coat or endosperm, but it is not possible for the embryo to directly communicate with these because it is surrounded by a cuticle that covers the protoderm surface. So, this function of communication is performed by suspensor. So, basically suspensor is like a transfer cell. It has different wall ingrowths or infoldings which increase the surface area for absorption, and once the nutrients have been absorbed by the suspensor, they are transferred to the embryo proper. Now we know that of course suspensor is involved in supplying or providing nutrition to the embryo. The primary source of nourishment of the embryo is the suspensor, and of course endosperm, a whole tissue, is meant for the nourishment of the embryo only. So, endosperm being the primary tissue, and then there is suspensor which is present. Suspensor is a part of the embryo, endosperm is a different tissue which is present in the seed. Presence of embryo sac haustoria suggest maternal sporophytic cells also nurture the growing embryo. So, endosperm nourishes the embryo, suspensor nourishes the embryo, and now we have maternal sporophytic cells like the nucellus which is present around the embryo sac, which also has a role in nurturing the growing embryo. Indirect nutrition of embryo is supplied by sporophytic cells, and this happens via the endosperm. Sometimes the growing embryo sac, it leads to the degeneration of the nucellus and the surrounding cells. Now, once the nucellus and the surrounding cells, they have degenerated, the content of degenerated cells gets incorporated into the endosperm. So, it is another channel for nourishment to the growing embryo. So, embryo, now we have understood that it derives its nourishment from endosperm, from suspensor, from maternal sporophytic cells, and of course with the degeneration of the nucellus and the surrounding cells, which eventually get incorporated into the endosperm and they supply nourishment to the embryo. In Euphorbia, Arabidopsis and Butamus, wall projections, they develop at the micropylar end of the embryo sac once the fertilization is over, and these wall projections are similar to transfer cells, and of course their function is to help in the absorption of nutrients. If you would recall, when we were talking about the structures of ovule, we did talk about epistase and hypostase. Hypostase, which is present just below the embryo sac, is a group of thick walled cells. Hypostase too is instrumental in supplying nourishment to the growing embryo. In glycine max, hypostase and the cells nearby, they have a two-way nutritional supply to embryo being operational in the embryo sac. So, these cells also supply nourishment to the growing embryo. Now, there is a very particular relationship which exists between the embryo and the endosperm. We already know that the growing embryo is dependent on the endosperm for its nourishment, right. So, it is like the embryo is dependent on the endosperm. In an embryo, the outer wall ingrowths of suspensor cells or the embryo sac wall, they are adaptations. What adaptations? They adapt to absorb nutrients from the endosperm. So, basically the embryo is absorbing nutrients from the endosperm. if you would recall that there are certain seeds which are endospermous while the others are non-endospermous. In non-endospermous seeds, all the endosperm has been consumed during the development of the embryo. And in endospermous seeds, of course, the endosperm remains even after the development of the embryo. The widespread presence of these wall labyrinths in the suspensor or the wall ingrowths in the suspensor, it suggests a transfer-like activity of these suspensor cells which we have been talking about so far, and these support a theory which is called the solute absorption theory for embryo endosperm nutrition relation. This theory was postulated by Schultz and Jensen in 1969, and it is only proving that the nutrition is absorbed by the suspensor from the endosperm. Right. Now, these wall ingrowths, the proliferations, whichever they are present, these basically increase the surface area and they support the short distance transport of the solutes, the metabolites from endosperm to the embryo. The plasmodesmata which perforate the suspensor embryo walls, they serve as a duct for this transport. Okay. So, I believe you have understood that what is this embryo endosperm relationship, and giving a quick recap, I would like to tell you that when the zygote is formed just after fertilization, it is a single cell structure, it undergoes divisions and eventually it forms the embryo. So, the first division is mostly transfer, giving rise to a basal and an apical cell. Depending on the number and the type of divisions in the basal and the apical cells, there can be different types of embryogenesis which occur in the angiosperms. So, if the basal cell will always divide transversely, whereas the apical cell has an option of dividing either transversely or longitudinally, giving rise to the different types of embryogenesis which are there. If the zygote divides not transversely but longitudinally into two cells which lie parallel to each other, that is a piperid type of embryogeny which is seen. Now, subsequent divisions in the apical cell and sometimes also in the basal cell, they lead to the formation of embryo proper. Basal cell rarely contributes to the formation of embryo, but when it does form the suspensor. Okay. Most part of the embryo is formed by the apical cell only by going further divisions. So, once a globular embryo is formed, there are different layers which are formed. There is an outer layer giving rise to the surface, and then the core gives rise to the cotyledons and the root like structures which are formed, and that is how embryo develops. This differentiation of different cells of the embryo is called as embryo patterning. Suspensor is very important in providing nourishment to the growing embryo, and of course, we did talk about the embryo endosperm relationship which exists. I hope you have understood everything, and thank you very much for listening patiently.